हम बात करने वाले थे तो बात करते ये बोल के और बता दो इसके बाद क्या बोल मैं वो बाहर आके बोलूंगी मैं बाहर है तो कान में बोलना है इसके सब कहानियां क्या बोला मुझे धीरे
पहले से पता है
देखिए सेम चीज वापस बोली जो मैंने बोली रो रहा था तो फोटो लिया बस इस बात पे लड़कों बोलती यही तो बात है रो रहा था फोटो लिया रो रहा था देखिए अब क्या अब बात हो गई खत्म ठीक है अब जो मेरा गुस्सा है ना खाली लेकिन मत करो मैं अभी सोकर उठी हूं और मैंने बहुत ही वियर्ड सपना देखा मेरे को मत बता दो इतना चाहते हां अरे ये आज बोला से आज बोला था मर्द है क्यों अच्छा ये प्रॉब्लम है अरे भाई तेरी वजह से मैं इन दोनों के बीच में फंस गई मेरा दिमाग अरे दोनों मिलके
मैं क्या किया परफेक्ट और कौन सा
वाली क्यों सीक्रेट्स रखता है जान शास्त्र तो बताता नहीं तुझे जानना है वो जाना से ज्यादा क्या बोलता है वो क्या बोलता है मैं इसमें बताने जा रही हूं अदर को मत बता वो बोलता है एग्जैक्टली बोलता है नहीं यार वो वही बोलता है मैं जानती हूं
नहीं ये तो नया के साथ ही बोलना वो
बिना के साथ बोलता है बारे में कुछ नहीं बोलता बहुत ही बढ़िया हां सुन ले था जाओ और ऐसे बता अरे कुछ भी मेकअप करके कुछ भी बोल दे भाई
सुन
क्या
कट्टी और कट्टी ना
तू ऐसे भी कुछ काम कर है जा
हां ठीक है कट्टी
हां जा
आजा जा
बैठ तू इधर आके बैठ
मैं कुछ नहीं किया
सब तेरी गलती
हो गया ऊपर
Oh my God! हमारी लड़ाई हो गई और मैं गलती से बीच में बैठी मुझे नहीं पता था इतना नोटिस नहीं करोगे मेरे को लगा कि कुछ बड़ा मैटर हुआ है ना चिंती मैटर
अमीर लोग 200 रुपए 2
फर्स्ट मैटर में हो गया तो ये तू जो मेरा तेरे को सारे कुछ किसके लिए अच्छा रोड में छोड़ने के लिए कौन लड़के गया था आज वो लड़कियों को हाथ छोड़ के गए थे वो उसका गुस्सा होना था ना ये लोग रोड पे तब एक बार आप देखना तो बहन और भाई देखी नहीं होगी तो ऑफिस पहुंचे तुम दोनों एक ही हो देखना मेरे यार ए यार ये मन नहीं रहा
अरे मेरे को पहले बायो की नोटबुक मैंने अपनी बनाई है लिखना क्या है बायो की नोटबुक रीडर बैठ ना बना
मैम ने बोला बात
नहीं भाई वो मैम बोलती है यार
कौन करता है इतना बड़ा बोल मेरे को
बनाया
इरेजर
इसको भी चाहिए वो मेरे पास भाई देखो पैसा ही जल्दी बना दे नहीं रहो
दो मजाक करना है ना तो इकरा के साथ कर
नहीं दिया
बराबर तू तो है ही पागल है अरे एक और सब अरे उसका वो बताया ना
तेरा
तो मेरा भी अरे
हां
तो ये गुस्सा ये खास वो तेरे को गुस्सा
इसको तो मैं बता रहा था, मैं बस में बोला था
इसको पता नहीं था क्या एटीट्यूड। अरे तो बता तो रहा था ना। अरे तो मुझे पाला मेरा सांस करना।
हाँ ये बात में ये वैसे करती है इसी बात में। हाँ वैसे ही वैसे करती है।
तो मैंने 2 मिनट नहीं बोला 2 साल का है 1 मिनट नहीं बता रहा हूँ हमारा होता है
ये स्कूल में तो किसी करती होगा मैं सबसे करती हो जाऊंगा ये करती हो गई
अरे मैंने क्या करा
ये मेरे को 500 दे रहा है 50 तो मैं जैसे भी करती हूँ तैसे भी करती हूँ ये तो पहले पैसे दे के करती हूँ कोई पैसे नहीं दे रहा है 1 रुपए पैसे मत दो
थैंक यू सो मच
देख मेरा भी बना दो भाई
मेरा अच्छा टमाटर बना
पहले मैंने नोट किया आज बना है
मम्मी है
जो मम्मी है इधर अभी पहले उसको बता रहे हो जैसे कट्टी रखी बता रही है
देखो नहीं बताया दीवार को बताया
ये बायो है आज
देखो बताया देखो
ये बायो है
बायो
बायो है
थैंक यू बायो
लाइव में बोल रहा है
एकदम कितने पेज छोड़ एक ही पेज 10 पेज अरे एक पेज कर से 15
ठीक है
क्या बोले अरे दो लेक्चर है मैं आंसर करूं ये ले भैया वेदांत मेरे बोल रहा है वेदांत ये पकड़ना पैसा देगा वेदांत 50 रुपए दे देगा
पकड़ ले तो तेरा हो गया
मेरा भी अच्छे से
पढ़ाई नहीं कर यार
बना के तो दे
अरे यार मेरा इधर से उसके मुंह में
अरे पेन ले लेना
मेरा पर उसका वो हाथ
ये बस बायो है देख
ये 730
मैं बस सिर्फ बायो है ना बोल रहा हूँ
और मेरा आईडी कार्ड का आईडी कार्ड को लेके जाओ सुबह किसका देना है मोबाइल नहीं मैम को बोलोगे क्या प्रॉब्लम है ए आई कार्ड दे तुला वेगळं इन्व्हिटेशन देऊ आई कार्ड दाखव कसं तुला ये वाले तो अटेंडन्स शीट जरा खेचायचा नाहीये तुला तो पण घे यू तेरे पास दो आईडी कार्ड है हां इथे दे तू लेक्चर नहीं था कल? यो, बरी? तुमचं प्रोग्रेस मला नाही माहिती. तिला लेक्चर मी फर्स्ट देखते. मैम मैंने एक्स्ट्रा क्लास किया था मंडे को और आप आके करना पता नहीं मुझे बोला था टाइम टेबल में लिखिए टाइम टेबल में लिखिए लिखिएगा फिर वो मैम बताएगी ये उसका मैडम मंडे को टाइम पर कॉल नहीं मुझे दे मी थैंक यू मला वाटलं चेहरा परत जर हसताना दिसतंय मॅडम देखते देखते सगळं वाचायला
Good evening. What was the last topic we were discussing? What was the last topic we were discussing? Photosynthesis, no? Photosynthesis, light phase, dark phase, we were talking. Light phase and dark phase. So, products we had studied. Products of photosynthesis we did study. Products of photosynthesis, events of photosynthesis did we study? Events of photosynthesis done. And stomata diagram only we have drawn. I think the mechanism I haven't explained. Mechanism I haven't explained. So what are we? What we are done? We are already completed with the concept of photosynthesis. In this first part of the chapter, this is an important topic. Please take care when you are preparing for this chapter. You will get questions on this. We have studied about chlorophyll. What is chlorophyll? What is chlorophyll? Second last. What is chlorophyll? Tell me your names also. At the same time, I get able to give your names. Chlorophyll.
So me, I'm
Bhavya. What is your? What is chlorophyll? Define chlorophyll. We have written no some answers down there. You have to learn how to write proper answers also. What is chlorophyll?
it's a green
photosynthetic pigment produced by chloroplast.
It is a.
Green color photosynthetic pigment produced by chloroplast.
Read nahi karna hai. You are supposed to remember answer. It is a green colored pigment produced by chloroplast. Then what is chloroplast?
Chloroplast is the organelle which produces the pigment.
It is present.
Which is present in the plant cell and which produces the pigment. What are the two parts of chloroplast we had studied? Granum and stroma. Where does the light reaction take place?
in the graph.
Where does the light reaction take? पता था? हाँ, थोड़ा से नहीं चलेगा. As we are finishing with the parts, please go through. By the month of October, you should be thorough with your portion. Before your school finishes the syllabus, you should be more or less thorough with the portion. Okay? So it makes no sense. Let me just give you 1 minute of gyan. It makes no sense. You are coming here after school, sitting here for 2 hours, and in the month of May, sitting for 6 hours, and then studying, finishing up portions. But still, why? What type of viral infection is here? Sit here, no? Why at the back you go? So now when you are coming here, see that are you understanding or not? First of all, make it a point. Yes, we are able to follow the topics, what is over. The topics are getting over, you are keeping it, and after 3 months when you open up and start studying, what explanation, what, everything's up, khatam. This way, you will not get that much of a benefit. Let me just tell you, to get the maximum benefit, whatever is happening in the class. I'm not saying that every day 10 standard, not possible, I understand. But in a period of some 4-5 days, find out time, read, okay, what was done in physics, what was done in chemistry, what was done in biology, this. Am I clear? Did I understand those topics? If not, then I have to, at least not this week, next week I can ask the teacher again. You can clarify your doubts. Actually, when you start studying, by the month of September, October we will say bye-bye to you. We are done. So by the time you start studying, then day, night, if possible at 12 o'clock also you start asking doubts. We get students asking doubts tomorrow exam, the day prior also. Don't do that. At least a month, 15 days before, you should be full set with your syllabus, full set with your portion. So do start now, start from now itself. Now, chloroplast, what is chloroplast? It's a cell organelle. Two prominent parts, stroma, grana. What is the site of light reaction? Grana. Site of light reaction, grana. One basic difference between light and dark reaction.
It happens daytime, night.
Duration of the day, if it is light reaction occurring during the daytime. Dark reaction can happen all throughout the day, not to write night time. One prominent reaction taking place in the granum. Photosynthesis to grow story. What was that word? Photolysis of water. What does that mean? Photolysis of water.
Breaking down of the water molecule to release hydrogen ions
And
oxygen.
oxygen. What happens to that oxygen?
Some of it is used for processing food, the rest is.
Some of it is used for breaking down food. A small fraction used for breaking down food, and the major part releases into the atmosphere. Process known as diffusion. Process known as diffusion. Which structures are involved in the diffusion of carbon dioxide? Inward diffusion of carbon dioxide. Structures. Which structures allow the inward diffusion of carbon dioxide in the leaf? What was it? What was it? Stomata. Stomata helps in the inward diffusion of carbon dioxide. What happens to the glucose that is formed during photosynthesis? It gets, again, glucose. Two pathways are there. We studied what were those two pathways for glucose.
And some of it is stored as starch, and the other is
Distributed
distributed to the rest of the plant in sucrose.
in the form of sucrose by, with the help of which tissue? That has to be mentioned. So this is the summary. What are those three events? The three events. Who can tell me all the three events? The three events of photosynthesis. First is
Absorption of
absorption of sunlight by chlorophyll. What is the second step? Conversion of light energy to chemical energy, and the other part is breakdown of the water molecules to oxygen. And the third step is carbohydrates. I again tell you, this 3 points to write, rather this 4 points to write, clear and correct, you get 3 mark questions. So it's very easy to pick those 3 marks, but still students cannot write all these 4 points properly. See that you have understood all these that I have been talking about. On the last day I had explained you all these. Is this all understood? Anything to clarify? Any concept here to clarify? We are going to start with the concept of stomata, opening and closing of the stomata. So, where is the stomata present and what would be the structure? How does the stomata remain open? Why does it close? So when we are talking about the stomata, if you remember the other day as I was drawing the section of the leaf, there also I showed you that if this is the section of the leaf, the uppermost layer of cells, these are known as the epidermal cells, followed by some 2-3 layers of cells, obviously for any type of a leaf, depending on what is the thickness of the leaf we are talking about. And then there will be the lower epidermis. Talk about the lower epidermis. Here in the lower epidermis, majorly we understand there are some bean-shaped cells, and these bean-shaped cells is what we are talking about as stomata. Two functions of stomata to write. What will you write here? Two functions of stomata the moment we are mentioning. One is the exchange of gases. What is the function of the stomata? Number one, inward movement of carbon dioxide and the outward diffusion of oxygen. That's the first function. What is the other function? Release of water in the form of water vapor. That's the second function. Release of water in the form of water vapor. So two functions of stomata to mention. Write down. Mention the two functions of stomata, then we are going to understand the mechanism. Why does the stomata open? Why does it close? Two functions of stomata, right? The first is inward diffusion of carbon dioxide and outward diffusion of oxygen. Inward diffusion of carbon dioxide, and outward diffusion of oxygen. Outward diffusion of oxygen. Second, release of water in the form of water vapor. Release of water in the form of water vapor. So that's another function of stomata. Now, those of you who had already finished drawing the diagram, is there anybody who has not yet finished with the diagram? If you have not finished with the diagram, we will complete it right now. A neat, well-labeled diagram is what you have to draw. So we are talking about the structure of the stomata, and we'll also have to understand what are the layers of the cells. So this stomata that we are talking about, first of all, you'll have to label certain parts. Check what are the parts you have labeled. This is this stomata is a plant cell. So what will be the feature of this stomata? It would definitely have a vacuole. It will definitely have a vacuole there, and apart from that, it will also have the cell organelle known as chloroplast, which already we had come across with mesophyll cells. Mesophyll cells, what was the reason that the rate of photosynthesis is much higher when we talk about mesophyll cells? What was the property we had come across? Mesophyll cells also have a large number, numerous stomata they have. That's the reason these are those group of cells which can show a higher rate of photosynthesis. Apart from that, what is this? Is a plant cell, so definitely it should be having the nucleus. It will be having the nucleus surrounding the guard cells. These are the guard cells that I've drawn just now. The stomata is not only composed of the guard cells, but it also has got some surrounding layers of cells. Now let us talk about some surrounding cells. What are the surrounding layers of cells near the stomata? What are they named as? First is epidermal. The first group of cells are what? Epidermal. How many of you have finished drawing the diagram? Is it epidermal, the first layer of cells? Subsidiary cells. The first layer of cells are the subsidiary cells. So if I'm drawing the structure of the stomata, if I draw the structure of the stomata this way, so and label the layers of cells. So first of all, I'm talking about what are these group of cells. These are the subsidiary cells. What are these group of cells? These are the epidermal cells. These are epidermal cells. And then we are talking about the bean-shaped cells as the guard cells. The bean-shaped cells as the guard cells. Guard cell has got the vacuole, and the vacuole will always contain what? The sap. We are going to learn something, some more properties about the vacuole now. And we are talking about also the chloroplast. Now, talking about the chloroplast. Now, keeping in mind that this is a plant cell and it has got the chloroplast. So, from this concept, you have to start. How does the stomata remain open during the day? Why? I'm not asking a question. Now, how does the stomata remain open during the day? How does the stomata remain open during the day? We are to remember about the role of the chloroplast. Now, try understanding. After that, probably you'll be able to write the answer on your own. This chloroplast, again, the concept remains the same like those mesophyll cells also. This chloroplast are organelles producing the chlorophyll. The reactions are almost the same, undergoing the process of photosynthesis. Now, since they are undergoing the process of photosynthesis, they are producing the glucose molecules, and the glucose molecules are getting dissolved in the sap. So I'm talking about this sap. This is the vacuole inside, and what is there inside the vacuole? The water and the minerals, and that is what we are talking about as the sap. So this chloroplast undergoing the process of photosynthesis, preparing molecules of glucose, and since the glucose molecules are constantly getting dissolved in the water of the vacuole, as a result, the sap is getting hypertonic. So I'm saying that what would be the features of the chloroplast to understand? What are the steps? The chloroplast undergo a process known as photosynthesis, and as a result, they undergo the manufacturing of glucose, and the glucose would constantly get dissolved in the sap, making the sap extremely concentrated. But concentrated with relation to what? Making the sap extremely concentrated with relation to the surrounding group of cells. Now, which are those surrounding group of cells from where the water supply? Let me understand that there are some xylem group of cells also very much. Where are those xylem present? Here, here, here. Xylem is scattered all over there. If this is the section of a leaf, I may understand, yes, the xylem would be present here. The xylem would be present here. The xylem cells are all in a, or almost in a sequence all throughout the section of a leaf. So if the xylem cells are nearby, the guard cells, then these xylem cells are constantly supplying water to this group of cells. Which cells? The epidermal cells, the subsidiary cells, and from here the water will also enter into the guard cells. But we are saying that during the daytime, since the process of photosynthesis is very high, during the daytime the rate of photosynthesis is very high, sap is getting concentrated. If the sap is getting concentrated, what would be the direction of movement of the water? The sap is now hypertonic. This is what we have to understand. The sap is getting concentrated with large number of glucose molecules as compared to the surrounding cell. This guard cell is more concentrated as compared to the surrounding cell because this surrounding cell is getting water. Only water. Is this cell undergoing the process of photosynthesis? No. So this cell has got adequate amount of water as compared to this. This cell is having a small amount of water which is constantly getting is dissolving a large number of the solute molecules. So the sap is constantly becoming concentrated. A term that you always should remember as hypertonic. When I say hypertonic, I am comparing two different types of cells, this and this. This and this, this and this. I am comparing the guard cell with the surrounding cell and I try to understand. Then if it is concentrated, Then how is this guard cell behaving? The guard cell will always have a tendency of absorbing water from the surrounding cell. So since it is hypertonic, then what is happening? The water movement is always in this direction. The water movement is always in this direction. The water movement is always in this direction. Yes or no? The water movement is always in this direction. Which process are we talking about? We are talking about a process known as endoosmosis. Osmosis. Hypertonic medium will always result in endoosmosis. So now we are talking about the concept of endoosmosis for the guard cell. So the guard cells are constantly absorbing water from the surrounding cells. After some time, what would happen? After some time, this particular vacuole, it will not remain like this. This particular vacuole is constantly becoming bigger. It is filling up water. This particular vacuole will constantly become bigger. The vacuole has become bigger. As the vacuole becomes bigger, now the vacuole keeps on extending, so much of water filling up, and the vacuole becomes bigger. Now, at this time, what happens? This particular layer that we understand of the guard cell, how does the shape of the guard cell change now? The guard cell would push these cells and it keeps extending. The guard cell pushes these cells and keeps extending this way. The vacuole is now more extended. What happens to this layer? This particular layer also will keep on moving inwards. It keeps on extending inwards. This thick layer, it keeps on moving inwards. This outer layer, this will first of all push this layer of cells and keep on becoming more and more curved. And what about the inner layer? The inner layer also now keeps on pushing inside. What is happening as a result of the extensive amount of water being accumulated during the daytime because of the higher rate of photosynthesis? Endoosmosis, and now what happens? The guard cells are, what is that word we are going to use? The guard cells are turgid, meaning they are extensively filled with water. The guard cells are turgid. Turgid means what? They have become swollen, tight, filled with water. Reason, they are constantly absorbing water by endoosmosis. Till what time will this process continue? As long as the rate of photosynthesis is high, all throughout the daytime. When is this process supposed to slow down? Maybe by 5 o'clock, 6 o'clock, 7 o'clock, when the intensity of sunlight is not suitable for the process of photosynthesis to start. Already I have given you a concept about the light and the dark reaction. You already know if the light reaction has to start, a specific intensity of sunlight is necessary. एक बार process शुरू हो गया है, then certain reactions can happen even at night. But now I understand at this intensity the process will not start, but it can carry on with the dark phase reactions till 11 o'clock, 12 o'clock, 3 o'clock, 4 o'clock. But maybe at 6 o'clock next tomorrow morning again the intensity of sunlight is adequate enough to start the light phase. So ये है सारे concepts to understand. So this particular space between the guard cells, what is it known as? This is known as stoma, and the stoma is now wider, and this is what is there all throughout the daytime. Is the plant benefited by this process? Yes. Now that the stoma is wider, the diffusion of glucose, the diffusion of carbon dioxide is even more. The diffusion of carbon dioxide is even more, and the rate of photosynthesis is even more. But now by evening, by 6 o'clock, concentrate. You will concentrate.
She is giggling for no reason at all. Randomly.
By looking at you.
No.
Now she is remembering something. Something must have happened in school today. Look in the diagram.
I was looking at that.
You look something. Don't look curry, na. कोई curry नहीं बनने वाला है. So now, when we are talking about this, when we look into this, so what are we understanding? What are the steps? See there I am writing, rate of photosynthesis will always lead to accumulation of glucose. More the accumulation of glucose, the sap is hypertonic. What is the other word we use? If I have to say that this is dilute, what is that word we use? ये तो हो गया hypertonic. तो इसको क्या बोलेंगे? The dilute medium, hypotonic. So the guard cells are absorbing water. Give reason. Why does the guard cell absorb water? Because they are hypertonic and the surrounding medium is hypotonic, dilute. So this is during the daytime and the rate of photosynthesis. But just the reverse would happen now that after 6 o'clock, after 7 o'clock, the reverse of this process will happen. So during the daytime, these cells have taken so much of water. They have become turgid. They have become tight filled with water. But after 6:30, after 7 o'clock, when the rate drops, no more glucose newly formed. There is enough water, but now the glucose concentration is now becoming lesser. New glucose molecules are not getting formed because the rate has slowed down. But whatever glucose was there right from the daytime and the afternoon, what was happening? Are they going to stay here? No, they'll be distributed also, no? So this glucose molecules, the concentration of the glucose now here becomes much lesser. So after a particular period of time, what would happen? These particular cells will have so much of water. Now they are becoming hypotonic as compared to the surrounding cell. Because during the daytime, greedily they had absorbed water. But after some 7 o'clock, 7:30, what has happened? Rate इतना drop हो गया कि new glucose molecules अभी ज़्यादा नहीं बन रहा है. So now next what? This is becoming a hypotonic medium and the movement of the water becomes just the reverse. Now the movement of the water is not this way, rather the movement of the water is now this way. The movement of the water is now this way. When at night, when the intensity of sunlight is not favoring the start of the process, some few reactions are happening, but then the rate of formation of glucose is much lesser. So whatever amount of glucose is getting formed is almost all getting distributed. So accumulation of glucose in a high concentration is now not happening. So there is no question why should this become hypertonic, rather it is becoming hypotonic. So what is the direction again I mentioned? The direction is this way, the direction of movement of the water is this way. If this is so, then what will happen? After a particular period of time, will the vacuole start shrinking? Yes. The vacuole was now becoming swelled up, distended. Now the vacuole shrinks down. Now the vacuole shrinks down, and this vacuole will slowly shrink down. The vacuole will slowly shrink down now, not like the hours of the daytime. The vacuole is shrunken down. Immediately as the vacuole is changing its shape and shrinking down, of course the guard cell also will not remain like this. The guard cell also will start showing a change. So what would be the change in the guard cell? Now the guard cell also starts becoming limp. The guard cell also starts becoming limp. The membranes, the moment the membranes are pushed inside, these membranes also start wrinkling. The membranes also start wrinkling. So this would be the change in the guard cell. What about this side? This would be the change in the guard cell, and the guard cells are now wrinkling down this way. What would be the change in the shape of the stomata? Again, remember, I'm saying that the stomata or the stomatal opening is now much narrower than before, but are we saying actually closed? No, it is not actually closed ever. It is never closed absolutely. We never draw diagrams to show that it is absolutely membrane to membrane touch. That never happens. But yes, much more during the daytime it is narrowed down. So now we are going to say that the stoma is narrow. This is what we are talking about as closed stomata. When the stomata has closed down, is the diffusion of carbon dioxide more? No, the diffusion of carbon dioxide is also much lesser. Release of water in the form of water vapor is also lesser. Oxygen release is also lesser. So quite a number of processes inside the leaf has changed. So this is how exactly the process of opening and closing of stomata happens. But now if I have to use alternative terms, I say during the daytime this is what has happened. But now if I have to talk about that, what then during the night? What then during the night? Is the rate of photosynthesis the same? No, the rate of photosynthesis is much lesser. What about the process of glucose accumulation? Is it the same? No, that also has dropped down massively. What about the sap? Is it hypertonic now? No, the sap also now is dilute, and now we would say the sap is hypotonic. So what was the direction of the movement of the water from the guard cell towards the subsidiary cell? During the daytime it was?
From the subsidiary cell to the
What is this process now? Exoosmosis. What is the difference? Endo and exo. If the water is moving inside the cell, endo. If the water is going out of the cell, exo. And now this word to remember, opposite. The cell is becoming limp. The cell is becoming shrunken. The cell is becoming flaccid. The cell is becoming wrinkled like this. A condition known as flaccid, and this is what we talk about as the closed stomata. Clear the steps and the terms. When we are talking about a basic difference between the closed stomata, open stomata with diagram or without diagram, will be asked the question. Let us very fast write down the stages of mechanism of opening and closing of the stomata. Mechanism is, it is there in your book in a small paragraph. You'll have to write your answer in points. In the volume also there's a small paragraph there, but you'll have to differentiate between the closed stages and the open stages. Let us write out. Take the question there. Mechanism, explain the mechanism of opening and closing. Explain the mechanism of opening and closing of the stomata. Explain the mechanism of the opening and closing of stomata. First of all, we will write down for open stomata. Write down, put ahead open stomata. Put ahead open stomata. Under that you write in points, point number 1, during daytime you will write in points, number 1, during daytime, chloroplasts of the guard cells. During daytime, chloroplasts of the guard cells undergo photosynthesis and produce glucose. Undergo photosynthesis and produce glucose. Making the cell sap hypertonic. Making the cell sap produce glucose. Glucose. Making the cell sap hypertonic. Next point, hypertonic. Making the cell sap hypertonic. Next point. The guard cells are hypertonic. Second. Guard cells are hypertonic as compared. Guard cells are hypertonic as compared to surrounding cells. Guard cells are hypertonic as compared to surrounding cells and thus absorb water by endoosmosis. Absorb water by endoosmosis. Surrounding cells. What is it? What is it? You read out.
Guard cell hypertonic as compared to surrounding cells and
thus absorb water by endoosmosis. Same point you continue next line. Guard cells become turgid. guard cells become turgid. Now this word turgid you may or may not find in your book. I don't think so this word turgid is there in your book, but you may very well use it in your answer. The guard cell,
Spelling.
TURGID. Spelling. Third one, the guard cells become turgid. Turgid means when the cell is tight filled with water. Okay, if I ask you, are you able to understand whether the leaves are fresh or wrinkled? No?
Yes, ma'am.
Yes? Morning, if you have, I think almost all of you must be having some potted plants. Or if you see the plants around also, when they are being watered, or in the morning, they are a bit fresh, tight filled with water. And the cells and the leaves look very fresh. But afternoon, they look wrinkled. They're as if crumpled. No, they are wilted. They are shrunken. So when we are talking about the why shrunken? Because loss of water. And why so fresh? Why do they look so and the upper surface of the leaf very shiny also at the same time? That is because turgid. Every cell is filled with water. That's why. The guard cells are turgid. And as a second point, now go to the third point. Guard cells are turgid. Now you write down the third point. Guard cells are turgid was a part of the second point. Now third, the opening between the guard cells, widen.
In between the
Guard cells widen. A condition known as open stomata. Opening between the guard cells widen, a condition known as open stomata. Next head you put closed stomata. Open stomata, a condition known as open stomata. Next, closed stomata. At night, first one, at night, rate of photosynthesis is slow. At night, rate of photosynthesis is slow. This makes the sap of guard cells. This makes the sap of guard cells gradually hypotonic.
just make the sap of guard gradually.
Hypotonic. Full stop. Next point. The guard cells are having a dilute sap. As compared, no dreaming. As compared to sap, as compared to surrounding cells, comma, guard cells thus lose water. Guard cells thus lose water by what to write? Exoosmosis. Lose water by exoosmosis. And become shrunken and flaccid. They are no more bean shaped, they are long, flaccid. FLACCID. Shrunken and flaccid. Last point. The space between the guard cells becomes narrow. क्या हुआ? Spelling FLA. ठीक है? The space between the guard cells becomes narrow. A condition known as becomes narrow, a condition known as closed stomata.
Yes, the artery becomes narrow.
Condition known as closed stomata. आया समझ में? किसी को कोई डाउट है? हाँ? Look into your volume, page 153. Try solving question number 6. And Joe, whatever we are discussing, you will mark those questions in your volume also. Question number 6, page 153. 153. Question number 6, 153.
Which of the following condition?
Yes. Which? Correct. That's an assertion question. That's an assertion question. Try solving. Sixth one is a
Diagram.
where sixth one is a diagram is.
I'm next page.
Next page. Diagram.
हाँ. तो क्या diagram base नहीं करना है? Number 6, page 153, which extends to page 154. आया समझ में?
Yes, ma'am.
Read. No, you read and you do for yourself. Solve karo. I give you 2 minutes. After that, I'm going to ask you the answer. Done? What is the question? Let me check. Let me see. बुक भी किसका बुक है? तुम्हारा बुक कहाँ है? हाँ? क्यों? You got your books, no? Then see, now that we are solving these questions, you will need your book. All of you should have your volume. How many of you are not having your book? This volume, how many of you are not having? This volume, how many of you are not having? Now, how many of you are not having? Why? Oh, इतना पढ़ा है कि book ही भूल गए. Every day this book you have to bring because we are going to solve all the questions from here. अभी उसके book से तुम कैसे करोगे? Okay, now let us read this question. You take them. You see. One of you give me the book this side. You have only one more. Now, see this question there. I asked you go through the question. Number 6. Which one of the following conditions are true for the state of stomata in case of in a green leaf shown in the given diagram? Large. Now, first of all, this diagram, this stomata is a closed or open one?
Close.
Large amount of water flows into the ground cells. Gaseous exchange is occurring in large amount. Large amount of water flows out. Large amount of sugar collects. Now here they are talking about this type of questions always you will get in your paper, the assertion type. So which is the correct option?
C, ma'am.
A is true and R is false. No, I'm sorry.
On the sixth question, that's the sixth question. On the sixth question, we have to do that question after the eighth one.
This is the statement. So now the statements which are correct, you check the statements there. Out of these 4 statements, the correct statement is what? Large amount of water flows out from the guard cell. Large amount of water flows out from the guard cell. So that is the correct statement. one is A, both as A and R are true and R is the correct expression. Seventh one, if you have to do this, seventh one, if you check, nitrogen is an essential element for the plant growth and is taken up by the plants. Reason, the soil is nearest and richest source of the raw material like nitrogen.
Both the search reason are true and
Correct explanation of the.
sorry, say it.
Nitrogen is an essential element for plant growth.
Also, is it
The soil is the nearest and the rich source of raw materials like nitrogen.
What is
Here
it?
A and R both are true.
Inorganic.
Number 7.
the
Nitrogen is an essential. In case of number 7, it will be both.
That's organic.
Ha. A, R is the reason. Correct explanation. Okay.
Why did it say inorganic nitrogen?
Why? Because nitrate is inorganic in nature. Nitrates, sulfates, phosphates that you are talking about, these are not compounds of carbon. So obviously these are not organic. These are inorganic in nature. No, these are as it is radicals. So you cannot term this as an organic compound as it is. This is not a compound of carbon. Compound of carbon is considered as organic. Now next is we look into a concept known as the starch test. The starch test, the iodine test. Now look into that diagram. Actually in the textbook there is one diagram for the leaf. So if you have the textbook, return to that page. Just check out which page is it. Which page is it? Just after the concept of stomata, you have the diagram there. Let me explain you here. Here, what are we talking about? To understand the iodine test, we have a plant. We have a potted plant. We have to start with a potted plant. Now, in this concept, the potted plant that we have, this potted plant, you'll have to, first of all, keep it in the dark. So what are we doing? We are first of all de-starching the plant. This diagram, which figure number it is, check?
5.2.
5.4. How many of you have the book? Just check. 5 point. 5.2. 5.2. This diagram, check all of you. What type of a question you are supposed to answer? What is the interpretation of that diagram? 5.2. In the new book, I think it's the same.
I have the old 6.4.
Actually, new book is 5.2. Okay, I showed you the diagram. All of you are clear which diagram I am speaking about? Okay, now here we talk about a potted plant. What are we doing in this experiment is, we take a potted plant and we are to go for a process known as de-starching. What is de-starching? De-starching means keep the plant in absolute darkness. We are not to talk about any artificial source of light also there, because for artificial source of light also minimum rate of photosynthesis happens. So artificial source of light also should not enter. For how long? For minimum a period of 48 hours, 24 to 48 hours. Okay, 48 hours, 72 hours also sometimes, but 24 to 48 hours minimum we have to keep. We can keep extended to 72 hours also. So what is the objective of de-starching? This particular plant had undergone the process of photosynthesis. The leaves have stored the surplus food in the form of starch. The leaves have manufactured food in the form of glucose. Remember, you should not write that the leaves have stored food in the form of starch. Ye galat hai. You have to write leaves have stored in the form of food in the form of starch. What is the need for de-starching? Let the plant utilize all the starch. The plant has to utilize all the starch in this period of time. Now, after this particular period of time, what do we do now? This same potted plant we have to take, and this same potted plant we again keep it under sunlight. Again keep it under sunlight. For how long? But we keep it under sunlight for minimum a period of 6 to 8 hours. Keep it under sunlight for a period of 6 to 8 hours minimum. Destarching, allow all the starch to get over, stored food to get over. Then why put it under the sun? Put it under the sun so that the leaves can manufacture new glucose molecules and store starch afresh. Now they will store the starch again. Now from these potted plant, we are to take one leaf and go through certain steps. What are those steps of the iodine test, the starch test we understand? The first step is boil the leaf in water. Why? Boil the leaf in water to kill all the cells. Students try to kill microbes. We are not talking about any microbes here. Never write this answer. Of course, we understand sterilization and killing microbes with hot water. That is different. This is a common answer that is always seen in the papers written. Kill the cells or kill the microbes in the cells. No, kill the cells. But why kill the cells? What do you think? Why kill the cells? But boil the leaf in water so that
membrane is a semicolon.
so that
Membrane is not allows a pass.
why should we boil in water? I say
So that
to
photosynthesis
does not continue. So that the process of photosynthesis, unless and until we are killing the cells, the leaf may be detached from the plant, but the leaf as it gets sunlight, small amount of chlorophyll, enzymes, proteins, everything are still there. In that process of experimentation, what is happening? Do we want still more glucose molecules to be formed and even additional starch to keep adding there? No, whatever had been formed here, that much only we want to test. During the period of experimentation, we don't want new glucose molecules to be formed or starch to be formed. So boil the water. Boil in water. Next is boil in chlorophyll. Boil in alcohol. Boil in alcohol. Why boil in alcohol? To remove all the chlorophyll again. To remove all the chlorophyll. But now why to remove the chlorophyll? What would be the problem if the leaf is green in color? We want the leaf to be little faded away. We want the leaf to have a pale color. Why? Because now that we are going to add some chemical drops that will show some reaction, expected that it should show some reaction with the glucose or the starch. So unless and until the leaf is of a faded light color, we are not able to see any color change. If it is dark green or the regular green color of leaf, that will not allow us to understand any particular color. First step, boil in water. Second step, boil in alcohol. And what are the reasons? I already told you, so remember. Now third is rinse in water again. Why rinse in water now? To remove the excess amount of alcohol. Because now that we put the iodine drops will start reacting with alcohol separately. So that we don't want, rinse in water. And then next is what? Put some iodine drops. What is the color of iodine drops that we understand? The color of iodine drops is basically orangish yellow. It is yellowish orange or say orange yellow. So when we are talking about the entire solution that comes in the bottle, it is towards the dark yellowish side with a tinge of orange. But the moment we are putting this solution here, what do we get to see? We get to see some blue-black patches. We get to see some blue-black patches, and the surrounding regions are brown. We get to see some blue-black patches and some brown regions. What does this blue-black patch indicate? Presence of starch. Whatever amount of blue-black patches we get to see all over the leaf, this indicates the presence of starch. And the surrounding brown regions, which would be comparatively more also at times, that indicates the absence of starch. So this color interpretation we have to always remember the presence. So what is the color formed when iodine starts reacting with starch? Blue-black. So this is a common question for 1 mark also. What is the color change when iodine starts reacting with starch? Are we testing glucose here? Why can't we test glucose here? In this experiment, why can't we test glucose? We are testing starch. Why can't we test
boil the leaf in water?
No. Something more already we have talked about in the fate of glucose. There I had already talked about immediately. Now that the glucose is formed by the process of photosynthesis, remember the process of photosynthesis is going on and the glucose is immediately getting transformed to sucrose. Then the state, I can say this way also, that the sucrose is more stable a state than glucose. So it remains in this form, sucrose, for a longer period of time, and the surplus may get converted to starch. So this glucose does not remain as it is for a longer period of time, immediately gets converted. So what will you write here? Why by the iodine test we cannot test for glucose? Why are we testing for starch? Because glucose immediately gets converted to sucrose and then starch. Glucose immediately gets converted to sucrose and then starch. That's the reason we are not able to test glucose here. We are testing starch here. So this is all about photosynthesis that you have to study as per your syllabus. And with this we come to the end of autotrophic nutrition, the nutrition in plants also. Next is we are going to come across nutrition in animals. We are going to talk about nutrition in animals, and that would be heterotrophic nutrition. But to talk about heterotrophic nutrition, we will again take up two examples. One, single-celled amoeba, and human, the most complex group of organisms. What is the similarity between these two? And what is the similarity? We have written examples also, remember. Both are, kaun bolega? Both are heterotrophic, undoubtedly. Holozoic. 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 Yaad hai holozoic? What did we learn? Kya tha features holozoic ka?
They had
Parts and pieces of food, and amoeba holozoic. Amoeba is holozoic. Human is holozoic. There's a big difference when I talk about the amoeba and the human. But then when it comes to method of nutrition, I'm saying that both are holozoic. Then what is the similarity?
They can
They
digest.
digest.
Inside their own body. You need a break now, or later, 5 minutes, 10 minutes at one go.
Latest 10 minutes.
Since she's not. And okay. Take a break, then again after the break we will continue.
नहीं मैंने बोला था दोनों होता है
दोनों होता है लेकिन वो प्राइमरी रीजन वो रहता है इसका गला
वैसे सुन के मेरा गला सूख गया सच में ये बात है मुकुल क्या है वो यहाँ पर मिला बड़ा अच्छा पढ़ा है
नहीं
समझना
हमें नींद
हाँ वो अलग से है
देखो हंसी भी आई अबे कविता का
हाँ वो अच्छा
ये नहीं तो पागल
प्लीज
सा
बाबा में अच्छा पढ़ा
बाबा बहुत मजा आता है कुछ अच्छा पढ़ाती है भाई तुम लोग श्यामा मैम से पढ़ के
भाई
देखो
हाँ हम
श्यामा मैम वाली
हमारी
पढ़
अच्छा
के
है
नहीं बस
यार वो लोग चले
अरे यार what I got फर्स्ट वाले दिखाई उनमें था सेकंड में दिखाई